Barbara wasn't interested in the ordinary. She always, throughout her career, was puzzled by and pursued things that people, it's not that they hadn't observed before, there's a history, but it was those things that people kind of were puzzled by and put on the shelf that fascinated her, and which she tenaciously then followed up to because she wanted to understand them. Hi, my name is Nina Fedoroff, and I took Barbara McClintock's beautiful genetic work into the molecular age. We isolated and studied the molecular behavior of McClintock's transposable elements, very commonly known as jumping genes, but at the molecular level. She worked at the genetic level, so she inferred things from what was happening with the pigmentation patterns in kernels and leaves of corn. And we actually got down to the molecular level so we could see what was happening at the level of the DNA. Barbara was studying the behavior of chromosomes, and in particular, chromosomes in plants. She began to see a very high frequency of what was then called unstable mutations. They were popping up in these irradiated plants at a very high frequency. But then she noticed that in some of the progeny, the chromosome breakage was happening at exactly the same place. That intrigued her because she'd never seen that. Generally, when chromosomes get tangled up, they break quite randomly. But here was a family of corn plants in which the breakage was happening at the same place. The really extraordinary thing that she observed was that in some progeny, suddenly the place where the chromosome broke popped up at a different site. That led her to a whole new investigation in which that movement of that site from one place to another, which she eventually called transposition, led to the whole discovery. I've called it the wind behind the sails of evolution, because that's where it all starts, in trying new combinations of genes, new positions, new regulatory sequences.